Thanks, Sergey. So I'd like to thank everyone for coming over here on a Tuesday afternoon. And for those of you that saw my talk here last year, I want to let you know that a bit of it has changed, but some of it is the same. So I'll go ahead and wake you up when we get to that part. So if you were here last week, you saw a similar slide from Charles Price. Plants have a lot of diversity in both form and function, and that's something that we're sort of used to exploring and we see a lot. But what I'm here to tell you about is the diversity with fungi, my favorite organism. So because of this diversity in plants, we see that we've developed a lot of diversity within these fungi. They can range from pathogens, in which the fungi is geared towards harming its host, to mutualists, in which both are believed to receive a benefit. We even see things like decomposers, which are operating outside of the system, but are incredibly important for ecosystems. Interspecific interactions can be classified according to this species interaction grid, in which we can really tell that they range a multitude of outcomes from mutualistic, in which both species are receiving a benefit, to predation and parasitism, in which one is harmed but the other is receiving a benefit. Ecologists tend to think about these interactions as occurring one piece at a time. For instance, most ecologists only consider pollination as the pollinator and the single host species. But the reality is many of these interactions are co-occurring within the same host and co-occurring within the same community at the same time. So really, to move community ecology forward as a field, we need to start considering multiple species interactions. Which leads to really my overarching research program question. How do ecological and evolutionary processes shape the interactions between microbes and their plant hosts? My research program is really broad. I think about a lot of different microbes and a lot of different systems. In the past, I've worked with mutualist pathogen interactions, and more recently, I've been working with mutualist interactions. Today, I'm not going to be talking about either my dynamic systems modeling or further experimental work I've got, and instead I'm just going to focus on some field work and some modeling work that I'll be doing while I'm here. So here's my obligatory outline. We're going to start with a bit of general background to get everyone on the same page, and then move into my mutualist pathogen work, followed quickly by the mutualist work, and ending with some broad conclusions that we can draw when we think about all of these interactions together. So plant-enemy mutualist interactions is an area of developing research. If we think about interactions that we know for herbaceous plant communities, we see that in the dark purple arrows here and the light purple arrows, we have a lot of research for a very few interactions. For instance, we know a lot about what happens with mycorrhizae root herbivores and foliar endophytes and foliar herbivores, and we have a lot about the indirect interaction between mycorrhizae and foliar endophytes. But we know very little. Less than 2 studies are encompassed by these orange arrows for all of the rest of these interactions. What we do know indicates that there's a negative effect of mutualists on general insects, and there's a positive effect of mutualists on specialist insects. But this is primarily driven by enemy guild. For instance, we know that aphids, which are phloem-sucking insects, tend to benefit really strongly from their interactions with mycorrhizal fungi, but really negatively from interactions with foliar endophytes. Additionally, there's a large amount of specificity and context dependency in these relationships. So what's going on in the abiotic world around them is really important for determining the outcomes of these interactions. However, when we look at this graph, we notice that there's a key group of plant enemies missing, plant pathogens. When plant pathogens are considered, we see much of the same results. Mutualists tend to lessen the negative effects of enemies on plant performance, and this effect tends to be driven by the below-ground mutualists, particularly bacteria and fungi. For instance, here, we have squash seedlings which have both been infected with the common fungal pathogen Phytophthora capsaicei. However, this guy here is associating with the mutualist, and you can see that he's much healthier and happier than this squash seedling set, which has also been infected, but it was chlorotic after only 4 weeks, meaning they turned yellow and stopped photosynthesizing, and dead by 6 weeks. So you can imagine that having a mutualist may benefit the plant in terms of its tolerance to enemies. Which leads to my questions for this specific portion of the talk. Do mutualists and pathogens interact to alter one another's dynamics? And what role are abiotic context playing in altering the relationship between host mutualists and pathogens? Plants are a unique arena to study these type of interactions. They associate with a wide variety of microbes, some of which we know a lot about because of their importance in agricultural systems, such as Agrobacterium or barley and cereal yellow dwarf viruses. But most importantly for me, they're experimentally tractable. They don't move. When I plant them, I come back and they're still there. Viral plant pathogens are particularly important. These pathogens cause only about 15% of all damage in agricultural grasslands, which doesn't sound like a lot, but it's really responsible for a huge amount of crop production and quality decline. For example, here we have an eggplant which is healthy compared to a series of eggplants which are infected with tomato bushy stud virus. While these are perfectly edible, if you see one at the store, you're unlikely to purchase it, especially in comparison to this. So we're seeing this production loss. Additionally, viral plant pathogens are responsible for about 47% of all emerging infectious diseases in plants, including some current ones which we're highly concerned about, such as a pathogen that's decreasing the populations of bananas and decreasing the population of wheat. Barley and cereal yellow dwarf viruses, or BCYDVs as I'll refer to them throughout the talk, are particularly important. They infect over 150 different grass species, literally almost every known grass species. They're luteoviruses, and they're transmitted by aphids, solely by aphids. That means for a plant to become infected, an aphid has to feed on an infected plant and then move to an uninfected plant. There is no other way for infection to occur. It's an important agricultural pest, and infection is systemic, meaning it goes throughout the plant, although the virus itself is only localized to the phloem. In the phloem, it causes necrosis, which means it kills the tissue, and disruption of carbohydrates, stunting the plant, giving the name dwarf, and also causing unique color changes in the plant, the yellow and the red that we see here. Now we're going to talk about the fungus. So as E.O. Wilson said, most of the land plant depends ultimately on one relationship, the mycorrhizae. It's intimate and mutually dependent coexistence of fungi and the roots of plants. So we're going to go into this in depth in a bit, but to give you an overview, mycorrhizal fungi are a below-ground mutualist, which are unique in that they are able to gather nutrients such as phosphorus, nitrogen, potassium, and even water from the soil, which would otherwise be inaccessible for the host plant, and exchange them for carbohydrates from the plant. There are a lot of different possibilities for this to be mutualistic, and there are a lot of different ideas about why. We're going to go into a few of those in just a minute. But to give us an idea of what it means to be a mutualist, oh, it's over here. So here we see a plant growing, and these are the roots of the plant. And we're going to talk a little bit about what happens between the mycorrhizae and the plant in terms of this exchange of nutrients. So here we've got the plant root and we've got the carbohydrates. These are the sugars that the plant is getting ready to offer the mycorrhizae. The mycorrhizal spore here has collected nutrients from the soil, so either nitrogen or phosphorus, water, and they're going to exchange these nutrients with the host plant for that carbon that we just saw. So here, the hyphae are coming out. They're going to go to the host plant. The host plant is going to offer up some carbon to the, or stop. Oh, well, we'll just keep going. So that's going to offer up some carbon to the host plant. The plant's going to make a decision. Is that an equal trade? Is that the amount of carbon that I want for this? Or are they going to make a decision about, nah, I don't really want this? If it's a good trade, they'll go ahead and make the trade. The carbon will come from the plant into the fungi, and the fungi will give up those nutrients. If they don't like the trade, the fungi may release itself from that plant, either move to a different root within the same plant or a different plant. So you can see that you're starting to develop a system in which you have cheaters and true mutualists. You have the plant vying for carbon or for nutrients from the fungi, and you have the fungi vying for nutrients from the plant. And so you've got a possible system of coevolution and evolutionary relationships shaping within all of these interactions. All right. So first we're going to talk about what happens with arbuscular mycorrhizal fungi, or AMF. AMF are important because they form associations with their plant roots, and they provide nitrogen and phosphorus for carbon. While they provide a little bit of nitrogen, it's important to remember that AMF are primarily associating with phosphorus. Additionally, they're the most common terrestrial mutualism. It's believed that over 80% of all land plants form these associations. We have fossils that date these back to the beginning of plants. So we know that they're long-lived associations, and they associate with primarily herbaceous species. In order to look at these interactions together, I have manipulated not only the microbes themselves, but I used two different host plants as well as the nutrients that thought to be involved, both the carbon and the phosphorus. To manipulate carbon, I utilized CO2 chambers located at the USDA Agricultural Research Center in Raleigh, North Carolina. And then at the individual level, I used two species, 2 levels of virus infection, 2 levels of AMF infection, and 2 levels of phosphorus infection. My host species were Avena fatua and Bromus rhodiosus. These are introduced Eurasian annuals that are susceptible to BYDV infection. They form strong associations with AMF, which make them incredibly useful for this type of experiment. In order to infect the plants with virus, it's as exciting as it sounds. You have to paint aphids on the plants. So what you do is you feed aphids onto infected tissue that you know to be infected for a period of 5 nights. This allows the aphid to go ahead and get the virus into its system. And then you paint the aphids onto the experimental plants and cap them and feed them for a period of 3 nights. After 3 nights, the caps are removed and the plants are sprayed with horticultural oil, which is a non-systemic pesticide that doesn't hurt either of the microbes but kills the aphid. You repeat this process for 7 days in order to ensure that all the aphids are dead before putting them back in the growth chambers. AMF colonization is slightly easier. In order to colonize the plants, you grow the plants that are going to be the active component with both spores and hyphae of the fungi, and the ones that are going to be uninfected, you go ahead and grow those with a sterilized portion of the same inoculum. This is in order to control for possible differences imposed by the soil texture. So when you include the inoculum, it's possible that that itself is going to stimulate a response. So you want to go ahead and control for that the best you can. In order to make sure that these types of microbes are both infecting the way that we want, you visually assess for mycorrhizal fungi. You can see a little hyphae here. And then you use an enzyme-linked immunosorbent assay, or ELISA, to test for the BYDV infection. Additionally, ELISAs also allow for the quantification of viral particles within a plant, something called viral titer, which we'll talk about in depth. So I'm interested in how these interactions are occurring not only within the host, but how they're related to these abiotic principles. And interestingly, we see that the virus is stimulating fungal infection in terms of colonization under elevated CO2 more than under ambient CO2. And the reverse is also true, that under elevated CO2, AMF positive plants have higher viral titer than AMF negative plants, indicating that there's some interaction between these microbes going on that's stimulating the production of one another. Great. I'm going to knock it into the biomass, and I'm going to have a science paper, right? Not quite. So first, we have species differences. Bromus rhodiosus didn't respond as strongly or in any significant patterns. However, Avena fatua, we see that when the plants don't have AMF and are grown under elevated CO2, they're much better than the plants. They have higher biomass than those plants that are uninoculated with AMF. So there goes my science paper. Instead, what we see is that we have this interaction where fungal performance is being increased by the virus, particularly under elevated CO2, and viral performance is being increased by the mycorrhizae, particularly under elevated CO2. But plant performance is not responding in the same way, and in fact, AMF is actually decreasing plant performance, particularly under elevated CO2. So to tie it back into our questions, do mutualists and pathogens interact to alter one another's dynamics? Well, yes, AMF is increasing viral titer, and yes, BCYDVs are increasing fungal colonization. And it's possible that abiotic context is playing an important relationship between host mutuals and pathogens, as elevated CO2 is mediating these interactions. So some follow-up work to these questions was done by my former undergrad, now very much a graduate student with Keith Clay at Indiana University, Brianna Whitaker. She looked at this and thought, well, I'm going to break it down into the nutrient component. And she did an honors thesis with me examining what happens when you vary the amount of nitrogen and the amount of phosphorus available to the plant. And you get some interesting patterns. When we look at phosphorus, we get sort of an uninteresting response. Increasing viral titer with increasing host biomass regardless of phosphorus level. Basically, bigger plants have more virus. Well, okay, that's fine. But when we start thinking about nitrogen, we see an interesting pattern such that there's likely a threshold effect, such that at low, medium to medium high levels of nitrogen, it doesn't matter really if you start adding more nitrogen to the system. Your viral titer sort of caps off, and your biomass starts to decrease as well. So there's much follow-up work on this to start teasing out what's going on. But as I said, it's not just mutualists and pathogens that are interacting. Mutualists are interacting with one another. So for this portion of the talk, I started shifting and thinking about what happens with mycorrhizal mutualists and other types of mutualists. So my questions, which were originally about mutualists and pathogens, shift to start thinking about what happens when you have multiple mutualists interacting to alter one another's dynamics, and how is abiotic context really shaping these interactions. So for this work, I started working with ectomycorrhizal fungi, or EMF. These are slightly different than AMF in that while they both exchange soil nutrients, EMF are primarily responsible for nitrogen exchange, and they associate with woody plants, which means that they only associate with about 2% of the population. However, they associate with almost all pines, oaks, willows, firs, beeches, and birches, so things that we're really familiar with. They form these distinct morphological structures on plant roots, and many of you have even seen the above-ground sexual components of these guys, so you've seen a lot of mushrooms around, particularly in this area. Bacterial endophytes, on the other hand, are sneaky. They hide within the plant needles, but they're important because they stimulate plant growth, both by producing phytohormones and volatiles, and it's thought that they enhance nutrient acquisition, primarily by fixing nitrogen. Additionally, they protect against disease and abiotic stress. But only a few of them are culturable, so until recent developments of new sequencing technology, we've not really had a great idea about what's going on with these guys. So much of the work that I'm going to talk about here is groundbreaking in that we've only recently been able to identify bacterial endophytes within these systems. So as I said, this is all relatively new, but in 2014, a lab out of Finland was able to isolate bacterial endophytes and did a unique experiment in which they were able to factorially manipulate what happens with endophytes and mycorrhizae when you inoculate the same plant. So this is slightly different from an ecological point of view in that they used Petri plates to grow their little pine seedlings, and they inoculated down here with fungi, and then up here with the bacterial endophyte, and looked at the differences in host biomass. So I can see a bunch of you sort of squinting your eyes. This is actually fairly common in forestry, but if you're an ecologist or a biologist, it's sort of a little bit weird. So here is the control, and you can see that no matter what happens, the control always does less. So these are all of our microbial treatments that we're going to get into in just a minute. So the control always does less good, if you will, when it's by itself than when it's with a microbe. And then the MB here stands for the bacterial endophyte. And we can see that there are definitely differences based on when the bacterial endophyte is alone versus when it's associating with a mycorrhizal fungi. And what's unique about it is that it depends upon the species of mycorrhizal fungi that we're talking about. So first, when we look at the Suillus here, we see that the Suillus alone does very well. And when you add the bacterial endophyte, we see a decrease in the overall biomass. But the opposite happens when you look at the Piscilis. So we see that alone, it does okay, and it doesn't really change that much when you look at the when you add the bacterial endophyte, but it does so much better than the Suillus. So this is suggesting that there are differences based on the different mycorrhizal fungi that are involved. So species matter. For me, I started working with Monterey pine to look at these types of questions. Monterey pine, or Pinus radiata, is a coniferous evergreen. It's relatively short-lived for a pine, which means it only lives to be about 80. It's native to the central coast of California and Baja California in Mexico. So it's only native to 3 locations along the coast of California and 2 locations off the coast of Baja. And while it's been restricted to these 5 populations, it's planted widely for timber. So it's unique for its ability to survive in these wide environments, but it's really restricted in its native range. These small isolated populations have started to diverge in response to selection pressures, and we see this in a number of different ways. I've illustrated it here with these pine cones. You see that the larger pine cones all come from populations that are on the mainland, but the smaller pine cones are starting to come from the island populations. We also see this with the bark that the trees have, and additionally, we have some worries. So recent cones collected from Guadalupe Island have produced seedlings that are albino. This is not good. Additionally, we know that the Monterey pine have different species of mycorrhizal fungi that they associate with depending upon their location. So this represents the relative abundance of a mycorrhizal family as from samples collected at Año Nuevo, Monterey, or Cambria. And while the details aren't important, it is important to know that these colors are very different. So this suggests that the three populations have all started to diverge in the populations of fungi that they're associating with. But maybe that's just a result of the fact that they also have different environments, right? These are three different populations. They're pretty far away from one another. And so early work in 2012 grew these plants all together in a pot experiment in a greenhouse and infected them with two very common mycorrhizal fungi extracted from these locations, a Wilcoxina species and a Rhizopogon species. And we see that pines that are from the islands, that are from Cedros Island, Guadalupe Island, when they're grown with Wilcoxina, the Wilcoxina do really well. They're highly stimulated. However, when Año Nuevo and Cambria pines are grown, the Rhizopogon do really well. And they do less well here with the island pines. This indicates that it's not only the pines that are benefiting from this relationship, but it's that the fungi have developed this relationship as well, and that the fungi tend to benefit more from different genetic species of the host. And this might be because of genetic differences in the host traits or the genetics themselves. So in order to determine what's going on when we think about endophytic bacteria in the same system, recently minted Dr. Wilson and I went out to 3 populations and collected samples from 5 trees within each population. So I collected ectomycorrhizal fungal samples, in which I collected root samples from each tree, morphotyped those samples, which means you just look at the fungi and look at what looks together, and then further sequence those so that we have identifications on them. And Emily went ahead and cultured and Illumina sequenced a bacteria that we could isolate from the pine needles. And this was all done both in my lab at the University of Mississippi and in Emily's lab at the UC Merced. So we know that these populations differ in environmental characteristics. Primarily, we see that here, Monterey, which is in the middle and will always be in green, has higher percent silt, whereas Cambria, which is in blue at the end here, has higher percent clay, and Año Nuevo tends to have higher percent sand available to it. And while they have differences in soil water content, they're all relatively similar. So this indicates that we're seeing differences in the soil structure at these locations. And when you bring that into the sites themselves, principal components analysis even picks up on that. So we see that Monterey tends to separate out from Cambria and Año Nuevo, and Monterey is primarily characterized by the percent silt cover, or percent silt texture. But does this translate to possible differences in the bacterial endophytes? So when we look at population alone, we see that population does not actually structure these bacterial endophyte communities. There's no differences here. Those ovals are pretty solidly overlapping. Although we know that there are 3 species, Enterobacteriaceae 3, Enterobacteriaceae 5, and Erwinia that tend to associate more with Monterey, and 1 species, Burkholderia 2, that tends to associate more with Cambria. And when we take ectomycorrhizal fungi, we see that there are differences of the 3 sites. Primarily, Monterey is different than both Año Nuevo and Cambria. And there are 2 species, Cortinarius 1 and 2, which tend to strongly associate with Monterey. But is this because of the population, or is this because of the environmental factors? And so when you try and pull out environmental factors, we see that once again, the bacteria just don't care. But it is important for thinking about ectomycorrhizal fungi. So the ectomycorrhizal fungi are really curing on the percent silt and the soil water content of Monterey. But maybe that's just because individual taxa are really responding. And so if you look at correlations between the individual taxa of the bacterial endophytes and the ectomycorrhizal fungi, you see that they're all over the board. So while the higher values are in darker blue, the higher correlation values are in darker blue, and the lower correlation values are in red, indicate that there's a lot of differentiation for ectomycorrhizal fungi. There are some that really like soil water content, some that really like percent clay. The bacteria look like they're associating more or less the same way depending on the different types. So what I want you to take from this is that the ectomycorrhizal fungi are really exploring different niches and interested in the different types of environmental characteristics, whereas the bacterial are more broadly thinking about things. But how are they interacting to alter one another's communities? This is what I'm really interested in. So if we use a constrained principal coordinates analysis, we see that mycorrhizal fungi significantly structure bacterial endophytes. And this is particularly driven by two species, an S. coronarius species and Cortinarius 1, which really pull this relationship together. In order to determine whether this is important for these species alone or whether they're cueing off of some environmental or population factor, I ran the analysis by constraining the analysis first to population and then to environmental characteristics. When you constrain it to population, we get a new player in this system, this Pizio, and we swap. So the coronaria becomes more important and the Cortinarius becomes slightly less if you believe p-values. And then when we look at environmental characteristics, we lose this system and we get this one being stronger. So we see that the population and environmental characteristics are more than likely playing a significant factor in influencing these relationships. So by taking out the response or the influence of the population, we don't strengthen the effect of the bacterial endophytes, we actually switch it a little bit. And the same happens with environmental characteristics. And unfortunately, we don't have the ability to be able to account for both population and environmental characteristics in the same model yet, although that's clearly something that we'd like to be able to do. So to tie it back to the questions, do multiple mutualists interact to alter one another's dynamics? Well, yes. ECM are important for structuring bacterial endophyte communities. An abiotic context is important for altering the relationship as it mediates the interaction, possibly strengthening it. So for those of you that have been asleep, it's time to wake up because this is the stuff that I'll be working on here. So as I said in the beginning, I'm interested in not only the ecology, but the evolution of these types of relationships. But I haven't talked about evolution at all. So here it comes. Nothing in biology makes sense except in the light of evolution. I was told once that you can't have an evolution talk without this quote, so here it is. Coevolution describes the cases where we have two or more species which reciprocally alter each other's evolution. It's likely to happen with close ecological interactions, such as predator-prey relationships, as we see with a fox and the hare, parasite-host, like we see with this fish and parasite isopod. Additionally, we see it with competitive species and even mutualistic species, like ants and their aphids, or even mycorrhizal fungi. Additionally, you can think about coevolution in terms of an equation. So you can think of it as a way to maximize both host and symbiont fitness. In terms of the genotype and as an interaction between the genotype of the host and the genotype of the symbiont, both of which are trying to do their best so that they're doing better than the other one. Mycorrhizal fungi and their plant hosts are really important examples of this. There's reciprocal selection. The plant wants to do as well as it can, and so they want to give the fungi the least amount of nutrients it can and just do awesome. But the fungi want the opposite. They want to take all they can without having to give up as much as they can. So this means that selection should start to favor the plant to recognize these beneficial versus non-beneficial fungal taxa. And we might expect to see variation in colonization, particularly among different genotypes of hosts, which might have different traits associated with them. Selection can be broken down to a couple different components. We can think about it as stabilizing if it's a strict mutualism, meaning that we're seeing trait complementarity because it benefits them every time. So the fungi are benefiting every time and the plant are benefiting every time, so that you're seeing a strict benefit. It's diversifying if no combination of the host and the symbiont is best. So it might favor rare genotypes to come into the situation, or it might be favoring speciation, so it might favor things to diverge away from one another. Or we can see directional selection, in which we know that things are operating on a continuum from parasitism to mutualism. So you might see the development of cheaters popping into this situation to take advantage of this asymmetrical benefit. And however, I just talked about them as being three different things which are all happening in separate systems. It's possible that they're all happening at once and to different degrees. Additionally, while it's widely accepted that coevolution has played a really important part in shaping these interactions in the past, it's unclear what the contribution of coevolution is happening now. So we know that there's a large diversification of mycorrhizal fungi in plants, but we don't know whether this is happening because of parallel evolution in response to these abiotic pressures that they're imposing on one another, or whether this is happening because of current coevolution. In order to determine this, we really need data from experiments which directly estimate selection on plant and mycorrhizal traits. And as much work as there's been done with mycorrhizae, this has never actually been done. So the goals of this experiment is to look at variation and colonization among different host genotypes, and also assess whether directional selection affects mycorrhizal relationships. And so the work I'm about to present now is preliminary. So if it will change, and don't hold me to it. In order to get at this type of experiment, it really is helpful to control for one of these factors. So I chose to control for the abiotic environment by growing everything in a common garden. Common gardens are classic studies that are designed to control for abiotic factors. So in theory, when you have everything in the same location, day length will be the same, sunlight will be the same, rainfall and temperature will all be the same. Additionally, for my purposes, biotic factors should be the same. So you should have the same potential for fungal colonization because everything's in the same location. To this end, I grew 12,000 plants from 43 different families in a common garden in Cambria, California at the Kenneth S. Norris Rancho Marino Reserve, which is run by UC Santa Barbara. The Kenneth S. Norris Rancho Marino Reserve is luckily in one of the only locations that still has Monterey pine, so you can see it coming down the mountain here. And so growing these seedlings there makes sense because the plants already exist there. So you can see some mother Monterey pine here, and my flags indicate the beginnings of rows, and there are 50 seedlings per row. So the goal was to have these guys in the ground for an entire year. However, as happens, it was the hottest summer on record. A large portion of these dies, and so a big portion of my fitness measurement is ability to survive hottest summer on record. I collected a large number of traits, including survival, biomass, root to shoot ratio, relative growth rate, which was assessed as the length of needle-bearing stem over time, so these were measured for the longest needle-bearing stem throughout the summer, as well as total root length. I also assessed a number of microbial traits, including raw number of tips that were colonized, morphological identities, which were further identified with Sanger sequencing, exploration type, which is how the fungi is exploring the soil to gather these nutrients, whether they make really long fungi that are going everywhere, or whether they're really short fungi that are only getting things that are really close to them, whether they like water, so whether they're hydrophilic, or whether they don't like water, whether they're hydrophobic, as well as colonization intensity. Are you getting just a few little bits colonized, or are you getting heavily colonized roots? So preliminarily, we see that colonization does tend to differ by host genotype. Right off the bat, we see that these mainland populations tend to have higher average number of tips than the other populations, except for Monterey here, which is our problem child. When you pull out the islands separately, we see that Cedros tends to have higher colonization than Guadeloupe, but they're still relatively low, especially compared to these mainland populations. However, where you get to see some interesting results is when you look at the interactions. So when you look at Cedros the cross between Cedros and Cambria and the cross between Guadeloupe and Cambria, which are the two island pines being crossed with the mainland pine, we see that an increase in fungal colonization, indicating that some of the traits that the fungi really like that are associating in this mainland population are being kept up in these crosses, and that's helping the colonization of the particular plant. So this suggests that we're seeing differences based on host genotype and particularly based on traits. But how does this relate to fungal or to communities? It doesn't relate at all. So this is a pretty ugly NMDS plot. And it indicates that there's really no clustering based on genetic relation or genetic background of the host. And so in order to assess the coevolutionary relationship between mycorrhizal fungi and their host, I'll be using selective source analysis. This should allow me to identify biotic, which are interspecific interactions with these microbes, and abiotic sources of selection. While traditional analyses are great, they tend to neglect the source of selective pressures, and they only measure instead total selective pressure. What I'm really interested in is the selection that's due to the mycorrhizal fungi themselves compared to the selection that's due to the abiotic environment. Being able to partition this individual fitness is important to me, so that's why I'm going to be using selective source analysis. And just to give you an idea of what that looks like, this is my only equation, so you mathy people, wake up. So in selective source analysis is unique is that it uses partial regression coefficients from a model which described the plant traits, which here are in blue and they're X1 and X2, which are co-evolving in response to two fungal species, each of which have their own single candidate co-evolving trait, Y and Z. So in this particular equation, we're thinking about the fitness of the plant as a function of the traits that are associating with the plant, the two traits that are associating with the plant. So for instance, overall biomass, how about root length and relative growth rate or something like that, as well as the fungi. So for instance, an exploration type, so maybe going out into the water is really important, or being able to go out really far into the environment is really important. Or being able to colonize really fast, so having a ton of colonizers right there. And you can pull out not only the effect of those individual traits, but their possible interaction with one another in terms of a plant and a fungal trait interacting, or their interaction with one another in terms of two plant traits interacting, or two fungal traits interacting. And you can even go crazy and start getting all of these broader scale 3-way and 4-way interactions. So this is what I'll be working on here, which means a lot of it's not done. But I couldn't resist playing with my data. So this is what we have for preliminary results from this work. It will definitely change. There's some things that are still wonky with this model. And once again, this only looks at what's happening with directional selection. I haven't yet gotten to either diversifying selection or stabilizing selection. So it appears that we have a positive correlation when we think about how plant fitness is being influenced by the plant performance traits and the ectomycorrhizal performance traits. So here in the Z space, we have plant fitness increasing as both the plant traits are increasing and the ectomycorrhizal traits are increasing. Okay, that's cool. That has an indicator we've got some coevolution going on here. But we sort of lose it when we look at what's going on with the fungal fitness. So here, fungal fitness is also going up in the Z space. But we've got a funky reaction in that fungal fitness is negative related to ectomycorrhizal performance traits and positive related to these plant performance traits. So there's a couple of things that could be going on here which indicate that I still need to work with the model. One, it could be my definition of fungal fitness. I'm using just the raw number of tips that were colonized as a definition. That might not be the best definition, but as you can imagine, measuring fitness in a fungi is a little bit rough. So that could be one issue. What I think is more likely to be the issue is that some of these fungi are probably, right now I've got exploration type as a fungal trait included within the model. It might make more sense to have multiple models for each fungal trait, and so I can pull them out that way. It could be that having these fungal traits is just having the multiple exploration types together is really masking the pattern of what's going on. Additionally, one of the things that came out yesterday when I was working with the data is that the fungal fitness model is and should be a regular old count model. It should just be a Poisson or some sort of variation of a Poisson. It actually fits a gamma distribution better, which is weird, and I need to think about that more, A, biologically, what that means, and B, if that's true, fix this model to reflect that. So there's definitely some playing that needs to happen with its data, but that's why I'm here. So just to wrap it up with these questions, so at the basic level, we're seeing differences in colonization among the host genotypes. But how is directional selection affecting these mycorrhizal relationships? For plant fitness, it looks like we're getting a positive correlation, and for fungal fitness, it looks like we're getting a negative correlation, but both of those are really to be determined. So to bring it all back together, how are ecological and evolutionary processes shaping the interactions between microbes and their plant hosts? One, in the first part of the talk, we talked about the outcome of the pathogen and mutualist interactions being shaped by not only the mutualist identity, but by the abiotic factors. Additionally, we know that specialists play an important role in shaping mutualist-mutualist interactions, and that coevolution is an important component of ectomycorrhizal interactions. So I'd like to take a minute to thank my funding sources, both the UNC Alliance for Graduate Education and Professoriate, NSF, which has funded me at both the graduate and postgraduate level, as well as the University of Mississippi Honors College, which funded one of my former students, Sarah Steele, with her help on the ectomycorrhizal fungi bacterial endophyte project, as well as labs at UC Merced and University of Mississippi. And with that, I'll take any questions. Thank you.